right, hello once again. I am Christian Stevenson with the Mississippi State University Extension Service in Hancock County, and I am joining you today to talk about genetically modified organisms or genetically modified crops. And I'm going to talk a little bit about the history of genetic modification of crops, as well as talk about some of the effects of having those genetically modified crops in agriculture. And I want to begin just really talking about the history of developing plants and the history of humanity genetically modifying our crops. And really that starts many thousand years ago with selective breeding. And selective breeding of a crop is just taking qualities that we see in the crop that we're growing, choosing parents that have that particular characteristic, and producing offspring that are going to have more desirable characteristics. And we have selectively bred plants and animals for thousands of years. Really what we're trying to do is just adapt that organism's characteristic in a way that's going to be favorable for us or desirable for us. And oftentimes when we do this, we're going to wind up with a population of animals or plants that are going to have fairly similar genetics. So they tend to have the same strengths, but they also tend to have some of the same weaknesses. And one of the limitations for selective breeding is we're really limited to the genes that are already in that population of plants. An example that I like to use for selective breeding, because I've had a lot of fun working with tomatoes over the years, is the tomato. And if you look at the picture there on your top right, you are going to see a native tomato plant from South America. And you can see that the fruit on that plant are quite small. They're not a color that you're going to expect them to be. And then when you look down in the bottom right picture, you see what we now think of as our modern tomato. And that plant, when the first Europeans came to the Americas, the modern tomato was really already there. It had been selectively bred for many, many years by the native populations to produce that really valuable, really tasty product. Another really good example of selective breeding in plants, and probably the most dramatic example, is the selective breeding of wild mustard. And wild mustard, or Brassica oleracea, has been selectively bred into quite a few different products that we can now find at the grocery store, all the way from kohlrabi, where we've selected for that really thickened stem, broccoli, where we want the nice flower heads, Brussels sprouts with those really nice side shoots, cabbages, big heads, kale for its leaves. And so we've selectively bred that same species of plant to produce a wide range of products. Again, that process takes a long time. So having to select individuals that kind of incrementally develop that quality that we're looking for in that plant. And that was the method that we used for many thousands of years. Another really good example for this is our modern corn. Everybody loves corn on the cob, but modern corn, we actually weren't certain where it came from originally for a very long time. We eventually figured out that the origin of modern corn is a plant called teosinte, which looks very different, has far fewer kernels. Those kernels are enclosed rather than being open. And certainly there's a lot of breeding that has gone into selecting that plant to turn it into modern corn. Now, while we've been selectively breeding for a very long time, as we got into the 20th century, we started using some modern tools to help us out to produce plants that have those qualities we're looking for. And one of the techniques we used was something called mutagenesis. And really what mutagenesis is exposing seeds or exposing plants to either chemicals or radiation in order to generate mutants that have desirable traits, or may not have a trait that we don't want them to have. And then we can take those plants and then breed them with other cultivars. So some qualities in plants that we can arrive at through mutagenesis include having larger seeds, having different colors, having sweeter fruit. And in the period from really about 1930 to about 2014, of course, the process is still possible, just done much less. About 3,200 different mutagenic plant varieties were released. About 70% of those as directly the mutagenic plant, and about 30% offspring that were then bred on with other plants for that particular quality. And about 75% of those species were crop plants of some form or another. Another 25% were the ornamentals or decorative plants, where we introduced that that quality through that process of mutagenesis. Right now, again, going back to 2014, so a few years ago, there are about 1,000 mutant varieties of major staple crops that are being grown worldwide. We don't generally do labeling or identification of these varieties, and these mutagenic varieties aren't prohibited by any organic standard. Some examples of some mutagen bred crops here in the United States, some varieties of rice that were selected for having a shorter height, some varieties of barley that were adapted for increased yield and cold resistance. There's a type of peppermint that is resistant to a disease called verticillium wilt. Some types of beans with increased yield. One of the examples I really like to use for this are the Star Ruby and Rio Red varieties of grapefruit, where we really got that really nice bright red color in that grapefruit through this process of mutagenesis. And it's really important to kind of think of that process of mutagenesis is really kind of a random process. We don't control what change is being made in the genome of the plant. We're just making it more likely that we're going to come up with a mutant, and we might get a quality that's something that we like and that's useful for us. So now, as we've gotten into the modern era, we have the modern science of genetic engineering. And the goal of this really is the same goal as what we have in that long history of selective breeding, in the use of mutagenesis, and that is to introduce qualities into that plant or characteristics into that plant that are going to be attractive for us. And there are several different methods of ways that we have approached doing this. I'm going to discuss those in just a little bit of detail. And what we really want to do is we want to start off identifying a trait of interest, something that we would like to introduce into that plant. And the first place we're going to look when we want to do that is we're going to look to nature. We're going to try to find a trait that is going to be there somewhere in another organism or a closely related organism. For example, if we're looking for a trait that's going to help a crop survive in a specific environment, we're going to look for organisms that naturally are able to survive in that environment. Or if we want to improve the nutritional content of a crop, we're going to look for plants that produce that nutrient that we're interested in. And a really good example of this is the plant golden rice. Golden rice has an increased amount of provitamin A, and the human body turns provitamin A into vitamin A. That can be really important because vitamin A deficiency is the leading cause of blindness, particularly among young people and particularly in the developing world. And so they found the gene that will result in this increased amount of provitamin A in maize, or what we're commonly going to refer to as corn. And then we can introduce that into rice, and therefore we wind up with the trait that we're interested in the crop we're interested in. So once we found that trait of interest, we actually have to find the part of that organism's genetic code that contains the code for that trait of interest. So we can either compare that with a plant that doesn't have that quality and look at the genetic codes and kind of see what's different and use that as a way to determine what that section of the genetic code is. The other way of approaching that is that we can use some scientific techniques, some laboratory techniques to actually delete or knock out sections of the genome that we're looking at until we actually see that that trait is no longer there. And that way that lets us identify the genes that we're interested in, the actual genetic code that we're interested in, now introducing into the plant that we're interested in it being in. And that is the process of transformation. So a genetically engineered plant that's been transformed, that's had that genome inserted into it, has been transformed, and we'll often refer to that as a transgenic plant. So our goals, we're going to take many copies of that new gene, place that into the nucleus of the plant cell. Of course, we need to do this with the cell still being alive and healthy. We want at least one copy of that to insert into the genetic code of that plant cell. And then as that plant cell replicates, then that, including the new gene, we're going to get replication of that cell with that gene now in there. So we actually go from just that single cell out to an entire transgenic plant. And there have been several different methods that of delivering that extra DNA into the nucleus of the plant cell and have been successful in producing a transgenic plant. For brevity, I'm really only going to talk about two of them. And the first one I'm going to talk about is using Agrobacterium tumefaciens. This is a bacterium that naturally lives in the soil. And I first learned about Agrobacterium as a plant pathogen. So it causes something called crown gall in plants. And you can see over there in the bottom right picture an example of what's going on there. The bacterium invades the plant through wounds, either in the stem or in the root. And this bacterium has a ring of genetic material that we call a plasmid, or the TI plasmid. And once that's inside the plant, that TI plasmid leaves the bacteria, actually enters the plant cell and inserts itself into the chromosome of the plant. Once that's inserted, that TI plasmid, those genes are actually turned on, and that forces the plant to make metabolites, basically that the bacteria needs for growth, and it results in that huge overgrowth that we call a crown gall. So we can use that same method to introduce a section of genetic material that we're interested in. So we take advantage of Agrobacterium's natural ability to insert things into the genome of plants. We take the genes from that TI plasmid that alter metabolism and cause disease. We actually take those out and we replace that with the desired genes that we want to introduce. We then use those to infect and actually transform plant cells that we're growing in tissue culture. And we've initially thought this really only worked in dicots, though we have gotten that to work on some grass type species or monocots as well. So here's just kind of a diagrammatic way of showing you how this works. Again, you have that little section of that plasmid that's going to be altered. We're going to change that, insert that genetic material that we're interested in, put that back into the bacterium, and we use that to carry that into the cell. That's going to be inserted into the genome of the cell, or the genetic material of that cell, and then we're going to grow that plant from culture to produce that transgenic plant. Another interesting way that we've used to insert genes into plants, and one that certainly seems a little bit more science fiction, is a gene gun. And we also occasionally will call this a particle acceleration or microprojectile bombardment. And you can use this on tissue culture cells or seedlings, just about any species. And how this works is we actually shoot the DNA into the plant cell. So where we talk about particle or microprojectile bombardment, the gene gun actually takes microscopic gold or tungsten particles, coats that with hundreds of copies of the gene that we want to introduce, and then when it fires it into the cell, and once it's inside the nucleus, the genes will dissolve off of that gold particle, and it can potentially insert into a chromosome. Then you can select out the cells that have been transgenic, have been transformed. What they usually do is they'll have some sort of indicator along with that that will help them select out the cells that have actually been transformed. These days, the gene gun uses compressed air to shoot those little microparticles into the plant cells. The old version, I think the initial version of the gene gun actually used a 22 blank to fire those little microprojectiles. So here's plant tissue culture. One of the really interesting things about plants is that they have an ability that is really quite remarkable. We call that plant cells being totipotent. Totipotent really just means that we can take that one plant cell, and from that one plant cell, we can regenerate an entire plant. So in plant tissue culture, we take some tissue off of the parent plant, place that in an agar growth medium, has all sorts of nutrients in there and some plant growth hormones, and you'll get those samples developing into tiny plantlets. So tiny, tiny little plants that start to differentiate. You can see some images of that up in the top right. You see the sort of the initial growth of those little sections of cells, which we'll call a callus. And then, below that, down in that agar, we can actually see those little plantlets forming with the roots and with the top of the plant as well, that can then be moved out into a growth medium and just grown like a normal plant, which is a really quick way to generate a lot of copies of a plant. Now, we've talked a little bit about some of the ways we've used so far to make transgenic plants, but if you pay attention to science news, there's been a fairly recently a major new advance in the possibilities of genetic engineering. We call that, you'll often hear it called CRISPR, because its actual name is quite long. It's Clustered Regulatory Interspaced Short Palindromic Repeats. So CRISPR is much easier to say in conversation. But CRISPR allows us some really neat tools to work with genomes. And it's derived from a bacterial immune response, so a way that the bacteria defends itself from invasion. And CRISPR allows us to cut out a section of DNA that allows us to remove some undesired trait or weakness in a particular plant. And we can actually take that and insert something into the void that's created, so we can actually insert a new piece of genetic material using these tools as well. And because of the advances they've made, they have basically other little sections of genetic material that allow those cuts to be made really precisely right where they need to be, or have genes inserted right where they want to be. And not only can we be much, much more precise with this, but it also allows us to target multiple genes all at the same time and just be a lot more efficient in what we're doing. And so CRISPR really has a lot of potential to kind of be the future of where genetic engineering in plants is going. So with all of that said, I want to spend some time now talking about what genetically modified organisms, I am frequently going to refer to them as genetically engineered as opposed to genetically modified. And really that is me wanting to kind of say how precise we are in what's going on with these plants. So there's a lot of complex science that goes in and a lot of testing that goes into the development of these plants. And I want to respect that. So I'm going to refer to it as engineering because I think it deserves that. Now, we hear all sorts of things about plants being GMO or not GMO. There are really 10 plants or 10 species of currently available genetically engineered crops here in the United States. And you can see a list of them right here, starting with squash and soybean in the mid-90s to more recently apple, a variety of apple that was developed in 2017. And you see along the way, of course, cotton and corn, papaya, canola, alfalfa, sugar beets, and potatoes. So what I want to do is just spend a little bit of time talking about what modifications or what these genetically engineered plants have been modified for. And one of the things that has been important for genetically engineered crops is virus resistance. If you've heard me talk about plant disease previously, one of the difficulties in dealing with plant viruses is, unlike something like a fungus or bacterium, there really is no application of a treatment that you can apply that will solve that virus problem for you once the plant has it. And so resistance is incredibly important when we're trying to manage viruses of plants, because it really is just our most important tool. In squash and zucchini, there are several viruses that affect that crop. Cucumber mosaic virus and watermelon mosaic virus, zucchini yellow mosaic virus can be really important diseases that can really seriously negatively impact production. And so what they figured out is they can introduce a part of the gene of that virus into the plant, and that will interfere with the life cycle of the virus and prevent that plant from being affected by those viruses. Now, this is not a particularly widely adopted crop. There are a lot of different viruses that affect cucurbits, like squash and zucchini. So there's, last I checked, there were only about 25,000 acres of these virus-resistant squash and zucchini grown in the United States. It's kind of unlikely that you're going to run into them. Probably a better example of virus resistance in a crop being introduced through genetic engineering is the resistance in papaya to papaya ring spot virus. So papaya ring spot virus is a very serious disease of papaya. Can make the fruit completely unmarketable. You can see a picture of that down in the bottom right. And it does eventually just kill the infected tree. And it's very difficult to control because it is transmitted by an aphid species, and it's transmitted in a way that we call non-persistently. So all the aphid has to do is just really feed once, and it transmits the virus. So even treating for the aphids may not be successful in preventing the spread of the virus. Though we could identify some resistance in some wild varieties of papaya, we weren't able to get that into a papaya that we're growing through traditional plant breeding. And so what was going on is that in Hawaii, the papaya growers were essentially just, once the plants in an area got infected, they were just moving to a new island where they didn't have that virus established yet. And then every time they would move, you would get a short reprieve, a period of time when the plants didn't have the virus, and then eventually it would show up. And that's just really not a sustainable model to protect against the disease. Even in Hawaii, you're eventually going to run out of islands to run to. By using a transgenic plant that incorporates part of that virus, again, the sort of the outside of that virus, that gives us resistance to the papaya ring spot virus, and really is responsible for saving the papaya industry in Hawaii. Another really good example of disease resistance as a result of genetically engineered crops is late blight of potato. Late blight is caused by the pathogen Phytophthora infestans, and it's pretty much the most important disease of potatoes. It has a really strong influence on history here in the United States. Um, and management of late blight is fairly intensive. You have to apply a lot of fungicides to get effective control. And they've tried to develop resistant varieties through traditional means, but those resistant varieties tend to not produce a very good potato. They just either don't taste good or they don't grow well. And so they developed a new variety of potato called Desiree. My French is terrible, but that was developed between the United Kingdom and the Netherlands, and used the genetic engineering tools to transfer the genes from those wild potato species that were resistant into the potato to provide that resistance. And really, was extraordinarily successful in providing resistance against late blight. One form of genetically engineered crops that I think people think of a little bit more commonly is herbicide resistance. Whether that be sort of the most commonly thought of is the Roundup Ready varieties for soybean and cotton and corn. And those are tolerant to the herbicide glyphosate that works on enzymes in the plant to make the plant unable to form amino acids. And really what's done is we incorporate a gene from a bacterium that produces a tolerant form of the enzyme that herbicide affects. And I'm not going to go too deeply into the plant chemistry that's going on there. But there are other forms of resistance that can be used. Another one is a different gene from a bacterium that allows that plant to just break down that glyphosate chemical rather than be affected by it. There are also herbicide-resistant plants for other chemicals like glufosinate, or the 2,4-D type herbicides or dicamba. And generally speaking, they tend to work the same way. They tend to come from a bacterium that's either going to change the target a little bit or allow that plant to detoxify that herbicide. Another really common use for genetic engineered plants, or something that we really think of very frequently at least, is their use in insect management. And this is a really interesting tool. What they've incorporated into these plants, particularly corn, is a bacterial gene that produces a protein that is really effective in controlling moth larvae. So little caterpillars, they eat the plant, they take that protein in, and it causes that caterpillar to die. This is a really effective tool because we're going to be getting those caterpillar populations very early in their development. And it's going to be compatible with biological control programs because we're not going to have to overspray that crop with the insecticides we would otherwise to get control of those caterpillar pests. BT, the chemical that is produced by that gene, is safe for humans and mammals and fish and birds. In fact, it really is very specifically targeted at caterpillars. So it doesn't do anything to beetles or to other insects as well. So it's an extraordinarily selective herbicide. In fact, you can go down to your garden store and actually purchase the BT insecticide. There are several different trade names for it, like Thuricide, or you can just see it as BT. and that's been available since the 1960s. And what's remarkable to me here is that Bt insecticide is actually right there in the organic standards as something that's commonly used in organic agriculture. Just another thing that we've done here, and I'm going to just kind of very briefly go through this, is increasing drought tolerance in some crops. So they found another gene in a bacterium that accumulates in cold shock bacterial cells, but really what they found is that they can use that as a tool to increase drought resistance in corn, and this can be particularly important in developing countries where we don't have as many options for irrigation, or where they may have problems with the amount of rainfall they get, allows them to produce corn very effectively. Probably the most recent one, this is apple, a form of apple that was developed in order to reduce browning. So when you cut an apple open, everyone knows if you leave that apple sitting out on the counter, if you only eat half of it, the apple winds up looking really brown and kind of unattractive. So what's happening is when you cut the apple, it's introducing oxygen into those plant cells or to the plant tissue that's damaged, and some enzymes in the cell of the plant oxidize, react with that oxygen, and produce that brown coloration. So what they did was just remove the gene, or what we call knock out or silence the gene that produces those enzymes, and so you wind up with an apple that's not going to turn brown. It can be really great, you know, for young people where they might get apple slices or something like that without having to deal with that browning. Of course, I believe the last one I'm going to mention is biofortification. And biofortification is just improving the nutritional quality of our crops in the case of genetic engineering, introducing a gene. Golden rice is the classic example of this, where the rice variety was developed to produce far more provitamin A than you would get in traditional rice, to produce up, again, 23 times more beta carotene than the original rice. And that means that somebody's going to be able to get their recommended dietary allowance in just 144 grams of that type of rice. Again, that can be really important because of the prevalence of vitamin A deficiency, again, particularly in developing countries. I do see a question here in the chat. Would it be true to say that mutagenic could never be organic? And so just to kind of go back to what I was saying at the beginning, or when I talked about mutagenic plants, it really is a separate thing. So mutagenic plants, or in using mutagenesis to develop plant varieties, you're again exposing that plant to some sort of mutagen, usually a chemical or radiation, and that produces that random mutation in the plant that may produce quality we like. Now that, according to the organic standards for that are set forth, those varieties are perfectly okay according to the organic standards. In terms of the organic standards and the use of genetically engineered crops, the organic standards specifically exclude the use of GMOs or genetically engineered crops, just kind of in their definitions. We can talk about the merits of that, and I will in a little bit, and I can go into more detail if people are interested in that. So there are a lot of concerns that have been expressed about genetically engineered crops, and I'd like to spend some time today addressing those, at least in brief, And again, this is an area where I encourage people to have questions. So when we talk about genetically engineered plants, one thing that's important to understand is that there really is a vast amount of information that indicates that these crops are just as healthy or healthier than the traditional crops that we've been growing. So what we want to look at here is the consensus of the scientists that research these topics, looking at major scientific authorities around the world. And those that have come out in support of genetically engineered crops include the World Health Organization, the United Nations Food and Agriculture Organization, and the American Medical Association, all having come out in support of the safety of genetically engineered crops. There have been, as of a couple of years ago, 1,700 studies on the safety of these crops. We reviewed 20 years of data, the strongest tests that we can possibly perform, over 900 studies in Europe and North American health data, and there has never been anything to indicate that these crops are not safe. They have the same nutritional composition as non-genetically modified crops. There are no links to new allergies, cancer, celiac disease, or any other disease. And probably the most important indicator that genetically engineered plants are healthy is that we've been eating them for 20 years, and there really have just been zero food safety incidents around that. So that's some really strong evidence in support of genetically engineered crops and their safety. They have done extensive feeding tests with genetically engineered plant products in rats and other animals, and generated no results indicating any danger whatsoever in consuming these crops. They've actually looked at it over generations of the animals that they're feeding, again, showing no effects in fertility or health of offspring, no effects on genetics, no effects on transfer of those genome materials from the genetically engineered crops. And a lot of times you'll see something on the internet, or you see something on a less reliable site, and it might quote something as a study. Very frequently, these things are not actually scientific studies. They're what we call anecdotes or anecdotal evidence, and they're not done in a research setting. And it's really important that we trust the science in this, in terms of understanding the long-term effects of these crops that have very, very thoroughly demonstrated that they are healthy. Often a concern that is expressed to me is that, yes, those studies have been done, but those studies are done by industry. And again, that's just not something that's the case. And so this information is pulled from a site called Genetic Literacy Project, which is a wonderful resource on not only genetically engineered crops, but a range of other topics as well. And it just shows the breakdown of different crops or different research projects and where their funding was. And you can see that government funding makes up a very large percentage of that here in the United States. Of course, that would be through the USDA. A large number of these studies are done at universities. Of course, there are industry-funded studies, but it's important that we understand that where it says industry-funded studies, that doesn't necessarily just mean that those are the companies that are making those products, but also competing industries and other organizations. The research has really been done by a wide range of different groups. Another topic comes up occasionally, a concern about the introduction of allergens. So an allergy is when you have an immune response or a hypersensitive immune response when you come in contact with a surface. And a lot of people absolutely do suffer from allergies. About 1 in every 13 kids has some form of a food allergy. And, we have seen some increases in allergies. 90% of those are caused by very common things like peanuts or tree nuts, milk or eggs, wheat, soy, even fish. Um, but according to the way these products are developed, before any genetically engineered or GMO product gets market approval, the actual protein that's being incorporated has to be compared to every known allergen. And then that whether it can act as an allergen is analyzed in comprehensive experiments just to demonstrate that no one's going to have an allergic reaction to this product. And in fact, there have been no GMO products that have been approved for human consumption where an allergen has been found. And one thing that we can look at is it's actually, in the future, quite possibly something we can do to be able to remove those common allergens or the factor in peanuts that causes that peanut allergy to arise up. So that's something that we can look at in the potential, not only for the benefit of the producers and the economics there, but being able to produce peanut butter that somebody with a peanut allergy might be able to eat. Another topic we frequently run into: herbicide resistance in the environment, farming practices that go along with herbicide resistant crops. One thing that we want to understand about that is that we're actually lowering the environmental impact of our farming through the practices that are associated with these herbicide resistant crops. There's reduced pesticide use. We're reducing or replacing more harmful kinds of herbicides with glyphosate, generally speaking. And glyphosate has a relatively low environmental impact. It is considered less harmful because it's not going to be as damaging to aquatic animals or plants than other herbicides may be. And it breaks down in the environment very, very quickly. So concentrations that enter the environment are going to be lower than the minimum concentration for that chemical to be toxic. And another thing that goes along with that, without having to get into all of the other effects of herbicides, is that one of the things that we can do because we have these crops is we can avoid tilling our cropland. So tilling really is a way to manage weeds in our crops. So, but when we till, we loosen the soil. We're going to wind up with more runoff. We're going to wind up with more of that soil eroding. That soil on a farm plot is going to be different. It's going to have higher concentrations of fertilizers, higher residual pesticides. We don't want that to run off. So removing that tilling process is going to eliminate a lot of potential harmful effects. And through the adoption of the herbicide resistant or genetically engineered crops, the increase in no-till farming has gone right along with that. You can see the graph there on the right, where the adoption of no-till farming and the adoption of genetically engineered soybean really just goes hand in hand. You can see on the left some data from the USDA's Economic Research Service showing the differences between herbicide tolerant and conventional varieties in terms, just, look at that blue section. That is the just no-till. If you look at the blue and brown section on that picture, that's no-till versus, as well as other conservation tillage practices. So you can see in that herbicide tolerant soybean, or in those, in the herbicide tolerant soybean, there's much more adoption of those conservation practices that have positive impacts on the environment. One thing we often don't think about when we think about agriculture is that we use machines to grow our crops. We use tractors. So the use of genetically engineered crops does reduce our CO2 emissions by reducing the amount of times we have to move that tractor around. So our biotech crops reduce CO2 emissions by about 28,000 million kilograms a year, which would be the equivalent of providing or removing 12.4 million cars from the road, or cloning, or just closing down 7.5 coal-fired power plants. So we are seeing environmental benefit through the use of these genetically engineered crops. Now, one issue that does require some thought is, particularly in herbicide-tolerant crops, the development of superweeds. So when we have the crops that are resistant to glyphosate, producers are applying more glyphosate, and we have seen an increase in some weeds that are going to be tolerant to that herbicide. And so it's important that we use some management tools in order to make sure that we are not developing these super weeds. That resistance can develop when that weed has a mutation that allows it to survive. It then reproduces and can carry that onto its offspring. So the periodic use of other herbicides in order to get rid of those weeds can get around that resistance problem. We can also use refugia, or areas of the field that are not sprayed, in order to make sure that we keep that population of susceptible weeds present there in the population. It was an issue. People believed that, that it was unlikely that we were going to see resistance to glyphosate or Roundup. And obviously, I mean, that has come to pass, and there was some over-reliance on that herbicide. When that is, when you are going to rely too heavily on one particular management tool, it is entirely possible, or even inevitable, that you're going to develop a plant that's resistant. So now to fight these resistant weeds, there has been the development of newer genetically engineered crops that sort of stack 2 or 3 different resistant traits to different herbicides, including glyphosate or Roundup, 2,4-D or dicamba, or glufosinate. And again, it's important to use those management tools to help maintain the susceptibility of those weeds in the population. Now, if you have turned on your television or listened to the radio over about the past year, you have likely heard something about glyphosate or Roundup and the risk of cancer. So the World Health Organization's International Agency for Research on Cancer classified glyphosate as probably carcinogenic to humans in a report that was published not too terribly long ago. There are some issues with that report, to put it very mildly. Some key information that would have supported the conclusion that glyphosate doesn't cause cancer was omitted from that report without any real good reason. Other studies that have examined the effect of Roundup or glyphosate and cancer risk have not found that result. Most notably, there's the agricultural health study that monitored the health of 90,000 people in Iowa and North Carolina for a period of 17 years between 1993 and 2010. And this group included farmers that actually applied the pesticide to their crops, their spouses. So the people who are going to be the most exposed are going to be the producers who are using these products. Among more than 54,000 pesticide applications, 83% of those contained glyphosate. And they found no significant increase in cancers among those who were exposed to the chemical. So that's a really strong argument that glyphosate does not, in fact, cause cancer. That was backed up by a 2015 study by the European Food Safety Authority that also concluded that glyphosate was not likely to pose a carcinogenic risk to humans. Another question that I've had that's come up is on genetically engineered crops and different species of butterflies, but often I hear this about monarch butterflies. There was a paper that was published in 1999 in Nature that concluded pollen from Bt corn could kill monarch butterflies. That study was entirely discredited. It was found that that was in fact not the case. But there, again, one of those things you pay attention to. There was a decline in monarch butterflies, and really that sparked the first discussion on food labeling in genetically engineered crops. Um, there were a series of articles that were run in the National Academy of Sciences, preceding the National Academy of Sciences for the United States, ran a series of articles looking at the effects of biotech corn or genetically engineered corn, specifically on monarch butterflies, and they found essentially no effect whatsoever. Going so far, Rick Helmuth, who's an entomologist with the USDA's Agricultural Research Service, told National Geographic, butterflies are actually going to be safer in a Bt cornfield than they would be in a conventional cornfield. What we want to think about there is that if you can imagine, of course, monarch butterflies are feeding specifically on milkweed. They're not feeding on our crop plants. So they're not getting directly affected by the crop itself. They're feeding on that milkweed. So if we get caterpillar pests or if we get other pests in our crop field, there's, without the presence of that insecticide control that we're getting from a GE crop, there's going to be some sort of insecticide application over that crop that is going to affect those monarch butterflies. So by having the genetically engineered crop, we actually avoid having to make that spray and actually increase the safety of the monarch butterflies that would be out in that field. Now, they have found a decline in milkweed in agricultural fields because we're controlling weeds in our fields a little bit better. They did find a 2010 paper was found that that showed milkweed populations declined dramatically on farms. So what I would say in response to this is certainly, we probably don't have as much milkweed as we would otherwise. I don't know that that's a significant thing that has actually reduced monarch populations. Most of that can be traced to weather events and a few other things. But my advice would be, plant a butterfly garden. Make sure that you're providing a place for those monarchs to feed. But keep in mind, and this is possibly my favorite slide of every slide in this presentation, that just because we see two things that happen at the same time, we cannot necessarily attribute those things as one being a cause of another. And so this is a graph of ice cream sales and shark attacks by month. And you can see they fairly closely follow each other. We don't see very much of either in the winter. And then as we get into July, there's a much increased effect of ice cream sales on shark attacks. But what's really going on here is that the weather is hot and sunny, and so people are on the beach. And so important that even when we do see that two things are happening at the same time, that we really look and understand what the causes of those things are. So one thing that I am particularly interested in is agriculture and biodiversity and sustainability. And so there really is a lot of information that demonstrates the impacts of genetically engineered crops on biodiversity at the level of the individual crop field, the farm in its entirety, and the landscape over a larger area, looking at biodiversity of crops, biodiversity of soil organisms, weeds, our land use, a wide range of topics. And the products that we currently have out have actually reduced the impacts of agriculture on biodiversity, which the real message that I'm trying to get across here is that genetically engineered crops are good for the environment. Through enhanced adoption of conservation tillage, reduction of insecticide use, more environmentally benign herbicides, increased yields so that we don't have to expand our agricultural land. All of these reasons, or for all of these reasons, genetically engineered crops have improved the environmental impact of agriculture. We can look at this looking at pesticide use. Certainly there has been an increase in the use of glyphosate, but at the same time we see less use of other herbicides. Seen an 11.2% decrease in use of insecticides, in adoption of herbicide, insecticide use in maize, while increasing herbicide use in soybeans. So we're reducing both herbicide and insecticide in maize, increasing herbicide use in soybean. And so again, we're seeing a benefit there. So you can see, again, herbicide use absolutely has increased. You can see that in a period from 1994 to 2010, that there has been a fairly significant increase in the herbicide, but it's important that we understand that the herbicide being used has less environmental impact. So direct comparison of those numbers doesn't tell you the whole story. But the other side of that is the reduction in insecticide use. You can see as we've adopted more BT corn, the rate of insecticide use has dropped dramatically. There was a recent, I believe it was an article in the New York Times that claimed that yields were not being increased by genetically engineered crops. And that simply is not the case. In the United States, corn and soybean yields have been increasing for quite a long time as a result of just improved genetics, improved practices. And as we've gotten into the adoption of genetically engineered crops, that yield increase has accelerated. We've seen better increases in yield through the adoption of genetically engineered crops. So what we want to think about really quickly is what we call intrinsic yield versus operational yield. So we can think of intrinsic yield as the actual maximum yield that crop could possibly get, which we're going to lose some of that intrinsic yield as a result of insect feeding or disease or weed pressure or a variety of other possible things. Intrinsic yield can really only be approached by changing the plant, because that's just the maximum that plant can do. What we're really interested in most of the time is our operational yield. That's how much we're actually getting after all of those other factors have come into play. So when we look at better pest and weed control as a result of genetically engineered plants, we can see that we are getting real benefits in terms of yield increases. And we see a lot of those increases in the developing world. We may see less of it here in the United States because we were already doing a pretty good job. So now we're taking the other side of the benefit, which is reduced labor and reduced environmental impact. But with Mexico, yield increases in herbicide tolerant soybean, 9%. I'm not going to read all of these, but Philippines, an average yield increase of 15% in corn. Hawaii, of course, virus resistant papaya made yields go up by 40%. India has a really striking story there with their insect resistant cotton yield, resulting in yield increases of more than 50%. And you can see these two charts here. The one on the left is a little bit kind of harder to parse out. But if you pay attention right there, where that red circle is, you see when we have the adoption of biotech or GMO or genetically engineered crops, and what we're really looking at is the rate of increase of that line, and you can see it get to be a steeper increase through the adoption of genetically engineered crops. If you look over at the figure on the right, that is specifically cotton yields in India. And again, you can see that really rapid spike there at the adoption of insect-resistant cotton varieties that really have allowed a massive increase in their yields. Now, one of the concerns that people have expressed is about what are called Terminator genes, or a seed technology that prevents the plant from being able to produce a viable seed. So you wouldn't be able to replant those seeds. The formal name for that would be gene use restriction technology, or GERT. While there was some work on this many years ago, no Terminator gene has ever been introduced. In fact, there's a quote there from Monsanto, basically giving their commitment not to use any of these Terminator gene technologies. And so what they relied on instead is contracts with producers that they will use those seeds and they won't save those seeds. Even though, generally speaking, saving seeds is not something that you see most large-scale producers do. One thing that kind of came around from this was sort of the rumor that the large biotech firms were suing different farmers for the use of the seed, or the crop would be cross-pollinated with that trait. And through no action of the farmers, and then the company would sue those farmers, and that's simply not the case. It's just never happened. One of the things or cases that are commonly cited about this is the case of a Canadian canola farmer, Mr. Percy Schmeisser. I'm not certain I am pronouncing that name correctly. And he did get into it with Monsanto for growing Roundup tolerant canola without paying for the technology fee, without getting that contract with Monsanto. Now, it's notable that the pollen for canola is can be transmitted by insects or wind, can move for a significant distance. And his testimony, or Mr. Schmeisser's testimony, was that his canola must have been pollinated by wind or blown into his field from a passing truck. And the test showed, when they actually tested his field, they demonstrated that 50 to 95% of his canola contained that gene. And it's simply not possible to get that much just through stray pollen or scattered seed. What actually happened is that he sprayed Roundup on 3 acres of field closest to a neighbor's field. They were using Roundup Ready canola, and a lot of those plants did have some cross-pollination, and those plants survived the spraying, showing that they had that resistance gene. He then selectively picked the seed from those plants and replanted it. So what he'd really done was basically, select out the seeds that had that gene, and then said that it happened by accident. It convinced the judge that Mr. Schmeisser did intentionally plant Roundup Ready canola. Now, the Canadian Supreme Court ruled that he had violated Monsanto's patent, though they did not award any damages because they said that he did not, because he didn't spray any Roundup over his field, he didn't get any benefit from the use of that crop, so he didn't owe them any money. Oftentimes when you hear this story, you'll hear a lot of the details left out, and so this is really a kind of an invitation to think critically about the stories that you hear when we hear about genetically engineered crops in the news. Not to spend a lot of time on this, but talk about genetically engineered crops and world trade. One of the things that you'll occasionally hear is, well, this product's not allowed in Europe, or this product is not allowed in some other country. And really that is, under the EU, different countries can block cultivation of GE crops within their borders. It's all done on a case-by-case basis. And several of their members, or a large number of their members, did vote to prohibit the cultivation of several different biotech crops. Probably the biggest country that does not allow either cultivation or importation of GE crops is Russia. That is officially banned. And although the European Union does not grow genetically engineered crops, it is one of the world's largest consumers of them. So they import quite a lot of them. More than 300 million tons of biotech corn and soy for livestock feed are imported each year, making Europe the largest regional consumer of GMOs in the world. And so this map kind of shows you where there are regulations restricting GMOs. Of course, you can see North America, there are not. The majority of South America, there are not. And then you kind of have a mixed bag in Europe with again, they import them, but they don't grow them there. And it's important that we understand that the action of the governments in these cases is not really driven by science, because the independent science organizations in each of those countries have come out with public statements that genetically engineered crops are safe. So some other factors that can go into the not allowing cultivation of GE crops can be just trade protectionism. There is pressure from activists in this. And probably the example that sticks in my mind, France has its image as a world food capital and wants to maintain that. And so while the science says that these products are safe, they're actually great for the environment, we have to get the politics there as well. One area where that comes up is the idea of labeling genetically engineered crops. And there is some argument that I kind of understand on this. Part of that is just, the pro view is right, just having a right to know that it's there. Having a tool to track problems is something that I think is really important. Generally speaking, I don't think making sure we keep animal DNA out of food for vegetarians is very important, because none of the crops use animal DNA that I'm aware of. I don't think many other countries are doing. It is a very good reason. And so, there is some arguments. There are some arguments that I think that are legitimate there. But there are also some negatives that go along with requiring labeling. And probably the biggest thing that really comes to my mind as a negative, or concerns me as a negative, is oftentimes having that kind of label has been demonstrated to sort of stigmatize the product or scare consumers. People tend to view it as a warning label, even if it's not a warning label. And unfortunately, that does, in many cases, kind of seem to be the intention of some of the people who are really pushing for labeling in genetically engineered crops. Probably the worst example of that would be Dr. Joseph Mercola. I'm not going to list his website because I don't want anyone to go to it. I personally believe GM foods must be banned entirely, but labeling is the most efficient way to achieve this. Since 85% of the public will refuse to buy foods they know to be genetically modified, this will effectively eliminate them from the market, just the way it was done in Europe. So, again, that sort of stigmatization, it's not based on science. And that is really what my concern is with labeling in genetically engineered crops. And here's my other concern with labeling in genetically or having that non-GMO label. Currently, there is a voluntary label that's used. And if companies want to use that, I am perfectly happy for them to do that. But occasionally, you are going to see that label on some things that don't really make a lot of sense. So the two examples I've pulled out here are Himalayan fine pink salt. Salt is sodium chloride. If it's pink salt, it's got some impurities in it that give it that color, but it doesn't have any genetic material to modify. It's just a mineral. Similarly, you have GMO-free water. And I don't even know what that means. It's also gluten-free, just in case that's another concern for you. And what I really like to go back to is I really think we need to pay attention to the science on this. And any time I have the opportunity to pull out quotes from two of my idols, I take the opportunity to do that. These are gentlemen who I respect a great deal, who have had something to say on the topic. The first of these is Dr. Norman Borlaug. Norman Borlaug was awarded the Nobel Peace Prize for his work in the Green Revolution in the late 1950s and early 60s, and is credited through his work for having saved 1 billion lives. And his quote is, "If you're a theoretical scientist, you can philosophize about this, but I've been in the field for a long time, and I believe genetically modified food crops will stop world hunger." Another gentleman I have a great deal of respect for is Dr. E.O. Wilson. He was chair emeritus of the biology department at Harvard, started the E.O. Wilson Biodiversity Project, is a native of the southeastern United States. I've had the pleasure of meeting him several times. And his quote is, "There doesn't seem to be any other way of creating the next green revolution without GMOs." And I want to bring this back to a concept I care very deeply about, and that is sustainability. Sustainability requires that the actions we take and the processes, particularly in my interest in agriculture, are good for the environment, or fulfill our economic needs, and maintain our social needs. So we need them to be equitable, profitable, and safe for the environment. And we achieve that by using the best technologies and the best science that we have. And so I hope you will